thank you everyone for your patience, as this is the first attempt at doing both face-to-face and a webinar. So thank you for being patient with me. And it is recording. So thank you. I'm just going to do a brief introduction. Unfortunately, Matt Gillingham can't chair this session because he's in another meeting at the moment, so you're left with me. This is the first seminar for the year, so welcome to you all for attending today, and it's good that we've got a panel to speak to you today. So we have Professor Martin Cole, Head of the School, Dr. John Carragher, a private consultant and industry liaison officer, both with AFW and SARDI, and Professor Maria Saarela from SARDI, an affiliate of the University of Adelaide as well. So I'm not going to talk anymore. I will let you guys get started. So hopefully it all goes
Thank you very much, Rebecca, for that, for helping us with the, I know we've got a kind of temporary IT set up, so I've got to talk into this and also talk to you guys. Pleasure this afternoon to spend some time with you on quite an important opportunity for South Australia, plant proteins. We figured it was a good kind of time to share where we're up to with it. It's a big opportunity. We want to get a lot of people around it. We're going to do kind of 10, 10, 10, or a little bit longer than 10 on each one. The good thing is, for me, I'm just going to give the big picture, and then these guys are going to get into some detail, and they're going to answer all the difficult questions. That's the deal, I think. So just in terms of the big picture, what I thought I'd do is just kind of put it into global, national and state kind of context in terms of some of the drivers that are really supporting this one. In terms of food security, you're probably aware, you know, and in my lifetime, we've doubled the amount of food that we've got around, but this time around, we've almost got to kind of, but that's come at quite a cost to the planet. Next time round, we've got to almost kind of do that again, but this time we've got to halve the environmental footprint. So things around sustainability, planetary health are very important. So we've got to, feed, nearly 10 billion by 2050 with diminishing resources at the same time, protecting the health of the planet and the health of us. So there's a lot of global work. You might have seen the Eat Lancet in terms of looking at, environmental metrics in terms of dietary interventions and things. And this is really what's driven, I think, a lot of the issue around meat and searching for alternative proteins. The global meat business is $1 trillion business. We've got to for us to double that, we can't double that in the way that we've been doubling it with meat, because we won't have a planet to live on. Our kids won't have a planet to live on. And it's really related to a lot of the emissions related to meat. So the meat industry, I should say as well that I'm not a vegan and I'm not against the meat industry or anything like that. I think it's important that we look at meat as our red meat business is a very important business for us, a $20 billion business. So it's not about being anti-meat or anything. This is about providing consumer choice around alternative proteins. So really in the marketplace, what's happening in the meat industry, so if you talk to the MLA folks and the people in the industry, there's some really good work to try and get that social license right. So a lot of, in this example here, is for the technology out of CSIRO, where you feed a small percentage of a certain seaweed, and that changes the lumen, the gut, the lumen in the cattle, and changes that and significantly reduces methane. Problem is, you've got to build a whole new business. You've got to build a whole seaweed farm, and there's some work there in Tassie and also in this state around that. And then obviously, so you've got meat without methane, or do you go meat without meat? So a lot of work to, can you recreate that eating experience, but without meat? Now, what drives the consumer? So I'm going to showcase some work that Wendy Umberger and the GFAR folks have been doing in this space. If we look at the whole social license thing, people are still buying food on terms of price, taste and quality, all of that sort of stuff. But you look at the trends, some of those trends are coming down, whereas the sustainability trend now is starting. So we call that the rise of the ethical consumer. More and more folks are interested in terms of the environmental footprint and sustainability around that. It's almost a bit like I think the market's ahead of the consumer as well. So if you look at a lot of the market and people, companies willing to pay for a reduced footprint, then you're going to be seeing more of that. But eventually the consumer will catch up. So these are some of the drivers. At a national level, we have a plan to get to a $100 billion farm gate by 2030. If we, on our current rate, we're growing, to get to 100 billion farm gate, we'd need to be growing at closer to 4%, currently growing at about 2%. So actually we've got to be picking some categories of foods that are growing faster in order to get to that sort of value. Interesting thing, I use this with when I'm talking to ministers and other things like that. Very often we talk about the value of agriculture, and then the food processing return to GDP is under manufacturing. Well, actually, if you put agriculture and food processing together, it's as big as mining or some of these other big sectors, resources sector. So, it's a hugely important business for Australia and hugely important in terms of the future prosperity. But how do we get into some of these new higher value areas? And just to make the last point there is, if you think there's been a lot of activity on the farm in terms of productivity and yield. A lot of the new developments, innovation will be through the value chain, I think, in the future. So these are some of the products. This was some of the CSIRO roadmap that we did. We've done some sizing of the price. So things like health and wellness is still the largest market driver out there in terms of food products. You're seeing this sustainable solutions emerging, and this idea of premium. This is kind of an experience and a premium experience. Again, all kind of examples of drivers in the marketplace around value adding. So let's just focus in. So we're here to talk about plant protein. So just in terms of, so the proposition is, we're already, Australia's already highly regarded for its red meat business. That's a protein business. And we have a very good position there. We're globally competitive and we command good prices, and we've got a trusted position. So the proposition is, could we, Australia, build a similar business off of plant protein? Can we double the protein business, but through the use of plants? That could be a 20 billion number. The protein landscape, it's been a really fast growing area, a lot of activity in new startups and ventures. I'll share one example from that space, but it's across different things. So think of non-dairy dairy, think of meat substitutes, drinkable meals. This is the meal replacement area, particularly where high protein, low GI is important to manage satiety and things like that. Even insect protein, I know we're doing a bit of work here. Kerry and others are doing some work. Insect protein, seafood, aquaculture is another big opportunity in terms of delivering protein, algal protein, and then you've even got mushroom, egg substitutes. So this area is a very active space, particularly in the startup world. And what you're seeing is actually a lot of these startups, if you trace where the money is, a lot of these startups are actually funded by other large globals. In some cases, a lot of the alternative protein startups are actually funded by meat companies. So Tyson are in here investing in some of these. So it's quite an interesting space. Just share this one quickly. This was one we did with Jack Cohen from Hungry Jack's, who approached CSIRO a couple of years ago and said, I know you guys are not really in that space, but what could we do if you were? So we instead of doing the normal thing that we would do in CSIRO, where you'd bake a load of IP, maybe 5-10 years or whatever, and then you try and find a market for it. We said, let's build a new vehicle around the opportunity. And what can we structure around that? So we put a new startup around this. We brought instead of doing a pitch to venture capitalists, we said, let's bring the venture capitalists in as part of the discipline with the scientists. And we call that venture science. So we try to kind of accelerate that process. It's been quite a success story. So after 2 years, they built a plant in Wodonga. They've taken on a lot of staff. Originally it was a product through the Hungry Jack's network, but it's now in retail, Coles and Woolworths and Drake's here in South Australia, and now exporting to 4 or 5 countries. So an example of what if you're building something around an opportunity, if you put the right minds around it, and you kind of work out new ways of working, then you can go quite fast. Here in Australia, in South Australia, you might have seen the state growth plan. Quite an exciting plan to grow the value of the agri-food and wine business from 15.2 billion to 23 billion by 2030. If you look at in that plan, there's $3 billion against plant protein. Now think of that. Our current wine business is $2.25 billion, something like that. So that's so imagine they're saying now this is a business that doesn't exist at the moment, really, largely in the state. So imagine we're now saying, well, we're going to have a $3 billion business by 2030, and it's going to be bigger than our current wine business. And really what we're betting on here is to be able to take, we excel in dryland agriculture here in the grains, but it's in the commodity business. So let's just take the fava bean as an example. Current fava bean business, it's worth about $160 million. The average price, 5-year price, $440 per ton. In a good year, like this year's a real bumper crop, but guess what happens, when there's lots of it, then because of price elasticity, the price drops out of the market. So we're now at $280, $300 per ton. So the proposition is if we could take the fava, let's just use the fava as an example, we could take a dryland crop and move it up into similar sort of value to beef mince or something like that. The delta would be about 20 times. So you could take a $160 million business and you could take it to $3 billion. And so companies like the startups I've just talked about, I think will pay a premium. So we'll move growers from a commodity business to a contract growing business. They'll pay premium for that particular qualities, and they'll pay premium particularly for carbon footprint and things like that. So the opportunity for us really then is to, and I'll finish on this one, really what we're talking about here is to, with our partners in SARDI and other players, is to build a whole of value chain approach to trying to help build this business. We have a fantastic opportunity. We have the fava bean breeding program. We've got Melissa here. So we have that, but that program, and Melissa, will tell you, actually that breeding program has not been targeted. It's been targeted mainly for yield and things like that. So, when you start now to take those market signals around protein content and quality and all of that sort of stuff, then you can start to now to, target the breeding program to a future business. We've got to, there's some things in the middle here where we've got to build some ingredient businesses too, because you've got lots of different applications in the food space, but we currently don't have a protein isolate, protein concentrate business here in Australia. So we've got to try and build that. It makes sense to do that next to the raw material. So we're in good discussions with a number of people to try and make that happen. But, the researchable questions along the way, there's quite a bit of research that we can do in this whole of value chain opportunity. And I think with that, John, I might hand over to Maria.
Yeah.
Hand over to Maria and John, who are going to kind of unpack some of the progress they've made on the science side. Thank you. You don't really need fun.
That might be interesting. Happy to have it. Okay, so I will sort of go back a little bit and tell about some basic aspects about protein. So protein is not just protein, like carbohydrate is not just carbohydrate. So what is important to think about is the quality of the protein. And this, of course, means that does it satisfy the metabolic demand of the consumer regarding amino acids and nitrogen. Bioavailability, how much of the protein that we eat can we actually take up. So it depends very much on how we process the protein. If we truly denaturate and kill it and do that, the bioavailability will go down. And of course, there are the food matrix and all that. Digestibility, and this is also impacted by the processing. So how much of the protein is digested in the small intestine. So bioavailability and digestibility are related, of course. biological value is a measure of how well the absorbed amino acid profile matches that of the requirement. And all proteins are not alike. And there is quite a complex way of looking at the protein quality, and it's called protein digestibility corrected amino acid score approach. I'm not an expert on this. I know it exists, it's a complex model, but this is how they try to model it, because it's not just that the protein is a protein, you have to figure out what the quality is. This is an example of values for digestibility of different protein. It varies. Typically, animal-based proteins have very high true digestibility, but also some plant-based proteins. What is interesting here that depending, for instance, if you look at the wheat, how it's processed, the digestibility varies. And it's the same protein, basically, same proteins there, if you compare cereals and refined and whole. So processing has a large impact on these issues, and we cannot forget it. So what about protein requirement? This is, of course, how much a consumer should, what's the intake level that will allow the nitrogen balance in the body. Currently, it's considered to be, this is done from WHO, their latest sort of consensus report. It's 0.6 gram per kilogram body weight per day. So let's say on average 50 grams. Typically, people consume twice the amount. So we overconsume protein already. And this is not contrasting anything that Martin says, because Martin was talking about the future needs and population growth and all that. If we think about the current population, there probably is globally enough protein for the population, whether it's the bright protein from the environmental viewpoint is a different thing. There are few consumer groups that are recommended to have higher intake, so pregnant and lactating women, and some disease conditions. When you have, for instance, an infection, it's consuming your nitrogen storages. But you don't need any kind of clinical nutrition. In short-term illnesses, you can sort of just deal with their normal diets. Elderly people are often considered to have poor nutrition. They don't as such have higher protein demands as adults in general, but their appetite might be, it's gone, they don't eat as much, so they need more nutrient dense foods. So in that sense, they need better foods and more protein. And especially if their physical activity is really low. What about if there seems to be some kind of weird idea that you can consume as much protein as you want and nothing happens? Well, this of course is not the case. Overconsumption has consequences. Like with any foods, they are not short-term consequences because of course foods are not acutely toxic, but you might see the consequences in 10, 20, 30 years. So, of course, it stresses kidneys because some way you have to get rid of your extra protein. Something will either it goes into colon, microbes deal with that, or it goes to bloodstream and kidneys deal with that. And if you have any kind of kidney condition, you have to be really aware that you don't overconsume proteins. So there has been quite a lot of discussion that are there any negative impacts on protein consumption? And this is difficult to study because people eat diets. You don't have people who eat just protein or carbohydrates. So how do you separate all these nutrients from each other? But it's obvious that anything that the small intestine cannot handle will enter in the colon. There you have your gut microbiota. Gut microbiota eats everything that you cannot yourself deal with. In the case of fibers, it's hugely beneficial because they produce short-chain fatty acids, and those are beneficial for the host. In case of protein fermentation, the metabolites are typically negative for the host. They are toxic compounds. And these toxic compounds link overconsumption of protein to colon cancer. So that's a clear route that has already been established. Of course, it's a risk factor, but it's a clear risk factor. This is statistics about protein consumption globally. And as we can see, the meat is here in red, that in the countries where they eat a lot of meat, the overconsumption is huge. And I was surprised that even countries where people are largely vegan or vegetarian, like in India, there seem to be, on the population level, enough protein. They are actually very close to optimal. Of course, there are then smaller parts of population who don't have this kind of optimal situation, but this is a generalization. So at the moment, there is no lack of protein. So we just need to figure out what kind of protein we eat. If we think about different protein sources, we have to think about what kind of amino acids they contain. Essential amino acids are something that human body cannot produce itself. So those must be achieved through the diet. And these are leucine, isoleucine, valine, lysine, threonine, tryptophan, methionine, phenylalanine, and histidine. And different plant-based materials have different limitations considering this essential amino acids. So pulses typically are limited in methionine, tryptophan, and cysteine, and cereals are limited in lysine and threonine. And due to this issue, it's important to have varied sources of protein so that you get as close as balanced amino acid composition in the proteins that you eat. So then going back to the discussion about plant versus animal-based protein, if we think about nutritionally, and if we only think about the protein fraction, we don't think about any other fat or anything else that, for instance, in plant-based material, it's clear that animal-based protein are the best. And this comes from the issue that the closer it is to our own flesh, the better it of course is, because it's the same. So meat proteins tend to be complete, they contain all the amino acids. Plant-based proteins are incomplete, so you have to then supplement or you use different sources. If we think about other nutrients in the plant versus animal-based proteins, so meat contains heme iron, vitamin B12, other B vitamins. Most plant-based foods don't contain these B vitamins at all. But then on the other hand, meat contains saturated fat, and dairy contains that. Plants contain fiber, phytonutrients, antioxidants, stuff like that, which you don't get from animal sources. So yeah, there's a balance. But if you think that plant-based protein is healthier than animal-based protein, it's not true, because the protein as such is only protein. The plant-based food is healthier because you eat the other stuff with the protein. You eat the fibers, you need the phytonutrients and all that. So if you hugely purify your plant-based protein, you lose all the benefits of plant-based diets. Remember that, and most people just don't seem to understand this issue at all, that if you only have a protein, it doesn't really make a difference. It's just a protein for the body. It doesn't care where it's coming from. So the problem with many of the current plant protein products, like meat analogs, is that they tend to be ultra processed. So the proteins are highly purified, their purity can be 80–90%. So there is no room for any other plant-based nutrients in that stuff anymore. And because it's quite difficult to make plant-based things to look like meat, you have to use all the tricks in your toolkit to make it happen. And that's why you need to think about processes, you have to think about ingredients, you have to think about additives. And none of this, if you don't have all these, you cannot produce these hamburger patties or whatever you want to produce. So of course, that's a point of discussion, that should we eat more natural plant-based foods and not try to mimic something else, because then you have these issues. Okay, this is one example. It's not an updated. We had another slide where we were looking just at means. So if you have only mince meat versus plant-based means, it's of course very clear that mince contains only beef. If you look at the burgers, of course, both with beef burger and plant-based burgers, the ingredient lists are longer, but it's typically much longer with this plant-based stuff, because you just have to do more things to make it look like a burger. Some of the burgers contain mineral and vitamin supplementations, zinc, vitamins, but for instance, this Beyond Burger doesn't contain any of that. So you have to be very careful that you get these things somewhere else than in your diet. Okay, I think that was my last one, then it's John.
Okay, thank you. I need those on now. All right, now to talking about what it is that we're doing on this campus. You've seen Martin's information about why we're doing it. There is absolutely market demand for it. You've seen information from Maria, essentially why we need to understand the role of protein better in our diets. But let's see what it is that we're doing on this campus to try and capture some of that opportunity. Well, those of you who know this campus well will know that around here we've got many decades worth of experience in plant breeding and agronomics. And some of that is in pulses related to this initiative. We've got, this diagram basically has boxes, two colors. The stuff that's in blue are things that already exist on this campus. The stuff that's in the orange color is still to be developed, and they're initiatives that we're working on at the moment. So we can take that raw material from the field, whether it's commercial material or from the research crops, and we can dehull it and we can mill it. If we stay with the blue lines, recently, last year, SARDI bought a machine that you'll see a photograph of in a few minutes, called a jet mill and a dry fractionation. This allows us to do protein enrichment. We can, again, with instrumentation that SARDI bought and some of the existing university material instrumentation, we can measure protein. We can measure fiber, we can measure the non-starch polysaccharides and the particle sizes. We can do sensory and technological properties like viscosity and solubility of the materials that come out of that. And we can then take them through into the new product development kitchen, and we can make them into baked products or drinks or something like that. The things that we were trying to add in orange include capacity to be able to do defatting and deflavoring of the pulses. Some of these do have bitter compounds. You can reduce that by pre-treatment, by heat and other processes, but heat is the easiest one. You can reduce some of the fat that would make some of the treatment more difficult. One thing that the university is trying to move into is the wet fractionation approach. This is something that many of you will have done at the laboratory bench scale. You make protein preparations, you do gel chromatography, or you do electrophoresis in order to separate individual products. That's on a very small scale. We're trying to do it on the scale of kilograms of material. Once you've got it wet fractionated, you take out all the things that you don't want, you then have to dry it. Then it becomes an ingredient that you can then put through the rest of the processes. One of the things that we'd really like to add to our box of tricks is an extruder, so that we can try to make those ingredients into the meat analogues, but it allows us to do other things as well with the materials. So we're not starting from scratch, we're adding extra boxes into capability and expertise that already is on this campus. If I talk about the actual processes involved in creating protein out of the starting materials, a lot of this was developed based on soy. So the techniques were developed based on growing soy in areas that were growing soy in quantity. Most of them are based on wet fractionation. And because soy has an oil component, it usually has to be separated out first. So you defat, you take that oil, and obviously it becomes soy oil. And then you do a lot of milling, you suspend it in water, and you wash away the things that you don't want, like the starch. Then you change the pH, you precipitate the proteins, you filter out the proteins that you want to capture. You then get rid of the fibers that you don't want. And at the end, you've got a solution that you then have to dry in order to produce the plant powder, the protein isolate. The other approach is very much simpler. It's called dry fractionation, and essentially you mill your starting material. As long as it doesn't have high levels of fat, you can take it straight to air classification and then split it into a coarse and a fine fraction. Most people think that flour, once you've made something into flour, you can't then further refine it. That is far from being the case. It's a bit like petrol, making petrochemicals. You can keep refining that material and even do it on a dry basis. You get a very different product at the end of each of these processes. Wet fractionation gives you about 75 to 88% protein in a concentrate or an isolate. And they're the sorts of things that you see at the health food shops. When you're doing dry fractionation, it's a lot less precise, and you get about a twofold increase in the protein level of your starting material. So it works, it's good, but you certainly don't achieve the same level of protein that you get from the other ingredients. Now, as I said, our interest in the wet fractionation approach through the university is a work in progress. We're talking to the chemical engineers about the sorts of equipment that's required and the area that it would take up, and obviously the costs of doing that. We're getting some very good independent advice from one of the engineers at CSIRO in Werribee, who have a very good plant of this type. So we're working on that particular area at the moment. As I said, we last year got delivery of the dry fractionation equipment. We've still got our L plates on as we learn how to use that particular piece of machinery. But I can say that as of last fortnight, we have some proof that we're actually getting enriched protein fractions from chickpea flour that we've put through that process. Still a lot of fine tuning to do, but we're starting to get there. These are the sorts of numbers that we're talking about with dry fractionation. So we've got a number of crops on the left-hand side, including things like wheat, starting off at about 12% protein. But by the time you do dry fractionation, you can get that up to 28% protein in that fraction. So that's a multiplier of 2.3. Again, with most of these, cowpeas 1.9, navy beans 2.4 times increase, And many of these are coming out in the 60% protein with mung beans and faba beans. We can go from 31% protein levels in faba beans to basically 70% protein in an air classified fraction. It's not the same as wet extraction, but there are advantages and disadvantages of this particular technique. One of those advantages of dry fractionation is shown here, and this looks at the energy and water requirements of the actual processing of the ingredient. So if we look, the yellow line is basically the starch and the product as it comes in, for in this case particularly is pea. We've got a bit of energy being used in the milling of that material. And then we've got air classification, which again uses energy. Some of this can be essentially renewably produced energy, so we don't have to get it from high carbon inputs. And then the air classification basically gives us a fine fraction, a coarse fraction, and of course there's going to be losses as the stuff gets stuck around the inside of the machine, as I'm very well aware. And essentially you'll see in the fine fraction, the proportion of green in that yellow has gone up because you're concentrating that protein there. Okay, let's contrast that with wet fractionation. We're starting with the same raw material, peas grown in the paddock. We have to mill it. It's got the same amount of energy required to do that as was in dry fractionation. But then we've got to solubilize everything and wash out the starch. So we've got all of this water coming out. We can keep some of that water and reuse it. We then have to change the pH to precipitate the proteins. We then have to get rid of that water as we precipitate the proteins and either filter them out or spin them out and capture them. We then have to stabilize those proteins and do another pH change. We're using more water again. And then at the end, we've got this spray drying step, which uses the biggest amount of electricity of all. So if we look at the environmental footprint of using this type of technique versus dry classification, you can see why SARDI has decided that for South Australian conditions, dry fractionation is probably the way that we want to go. We don't have enough of the water, and we've got expensive power unless you're generating it yourself to do these sorts of things in this particular environment. If you're somewhere else, you need to look at those local conditions. This looked much better on my computer than it does on the screen. It's basically some really nice work done by the Wageningen people that compare the technological and nutritional and environmental performance of faba beans, proteins produced by wet extraction and dry extraction. I'm trying to focus on the stuff that's been highlighted. FPR in their system is the air classified material. FPI is the isolate produced by wet chemistry. So clearly we've got a lower protein content, 64% compared to 91%. We know that that's going to be the case. That's just a function of the technique. The air classified material demonstrated superior functionality, higher protein solubility, and that's what's shown in that top right diagram. They're the solubilities of the protein at different pHs from the two types of material. So it was 85% soluble compared to 32% at pH 7. The foaming capacity was greater for the air fractionated material. That's shown in this little diagram in the bottom left corner. But the protein, the stability of the foam wasn't different. Almost certainly those benefits are due to the carbohydrates. The starch has a role in both of those functions in terms of how that material is going to be forming a foam. It also has gelling abilities, which makes it better for making patties and things like that. But the potential nutritional benefits of the aqueous process, you get higher in vitro protein digestibility and lower trypsin inhibitor activity. And also you get less of some of the anti-nutritional compounds that you can find in faba beans. Just to look at some of those figures here. So we're not saying that one technique is necessarily better than the other. We're just saying that both techniques have their place. They've got advantages and disadvantages. So with this one, we're looking at protein digestibility, the amount of, with pepsin after 1 hour, protein digestibility goes from 5.6% up to 6.4% if you're dealing with the isolate. If you're looking at the trypsin inhibitor units here in the last column, 3.77 compared to 0.33. So essentially you're washing away the trypsin inhibitor fractions or components that are in that flour. If we look at the anti-nutritional compounds visine and convisine, essentially in the isolate, they're non-detectable. So you can essentially eliminate that from your product if that's important for you. There is a proportion of the population that has a enzyme mutation that basically means that faba beans are potentially toxic to them. So it can be a different, an issue for some people. All right, the rest of the talk I'm going to just focus on dry fractionation, and we don't see a lot of bean seeds around here. We see a lot of grain seeds that have endosperm. These things really don't have an endosperm. They've got cotyledon cells, and they form the majority of the inside of the bean. The seed coat is mostly fiber. The cotyledon cells contain starch and two types of or major protein types, globulins and albumins. If you put it under an electron microscope and fracture the cells, you see this beautiful pattern with the big circles or big ovoids. They're the starch granules, and essentially between them are the protein granules. So it's a much, much smaller unit, and that's essentially what we're trying to do. We are using the fractured planes to try and separate the starch granules from the protein matrix that's in behind it. This is our new machine. This is where we lent it to the guys from Breaking Bad before we took delivery of it. And but this is the Comex air classifier that we purchased and was delivered last year. The material starts on the left-hand side, and you can see there's a hopper that takes delivery of the flour, the coarsely milled flour. The first unit on the weighing bench is the jet mill, so we can mill it to different levels of fineness in order to try and get the best separation of those starch granules from the protein granules. From there, it then gets sucked along. There's a dirty grate vacuum cleaner here at the end, which maintains the process flow. It gets sucked along to the next unit, which is the one with the green box on the top. That's the air classifier. And that splits the flour into large particles and small particles. The large particles fall underneath the green box. The small particles move into the next cyclone and are collected there. Why does an air classifier work, or what does the principles? Basically, I've tried to show it here. You've got a mixture of different sized granules coming into the feed line, and within your chamber, which is sort of air blowing around and keeping everything in suspension, you've got a hamster wheel with slots in it, spinning around at different speeds. Anything that's going is small enough and fast enough to get through the slots will go up the pipe and get carried away. Anything that's too big to go through the slots will fall down into the coarse bucket. So essentially, by adjusting the speed of the vacuum or the suction going through, as well as the speed of the wheel, you can change the size particles that are coming out through the fines. And that's what this graph here tries to show. Essentially, at different rotor speeds, from low speeds to high speed, you can essentially shift the particle sizes that you're capturing in the fines. At the end of the day, what you want from your fines is a food ingredient. Then the food technologists can get to play. We can turn that ingredient into all manner of things. Drinks, snack bars, lots of different baked goods, bread, muesli, pasta, protein puff type snacks, custards, as well as the imitation meat products, including tuna and chicken. So that's where the technologists play and the nutritionists have heart attacks. So our work plan, and this is my last slide, this is basically what we're doing at the moment, is that we're working on fava beans, field peas, chickpeas, and lentils. We also want to work with something like oats. Oats we can mill, we can actually classify out and enrich fractions with beta-glucan. We need to work on deflavouring. We need to work on whether or not you need to de-hull your material before you put it through this process. Or will it just fall out with the coarse fraction and you can save yourself a process? Do we need to defat under certain circumstances, and how we do that in a environmentally sensitive way? We are interested in the functionality of the fibre-enriched fractions, and we're interested in the starch and the fibre-rich fractions because they're valuable too. We're also working on trying to get that wet extraction facility up and running. And that's where we'll leave it.
Thank you, John, Maria and Martin. We will take questions. For those of you that are on the webinar, if you have questions, can you please type them in the Q&A box and I will work my way through those questions. But do we have any from the audience to start with? Yes?
A question for Martin. You showed the following example. Is a ranked system okay to ensure continuity of the supply process in terms of volume and quality?
That's a really good point. I think most of the players in the on the food side of it really want to try and diversify the ingredient base. So I think the story is so in where you can we can't grow soy here. Okay, it's mainly a northern crop. So there's a big piece around soy. We can grow fava here, but even things like hemp. If you take de-oiled hemp and then what you've left in the cake, that's quite rich in protein. So I think it does need to be a whole of a diversified strategy, because if you have one, I think the point you're making is if you have a fail in one crop, then you don't want to be So I think a lot of the manufacturers are trying to devise systems that can cope with different ingredient supply. So you've got that robustness and resilience in the system. It's a great question.
So they buffer between.
That's the intent. Good question.
Can I get the three of you to come up so the people on the webinar can actually?
I have to leave. I have another meeting starting at 1, so I'll be
I'll
leaving in
just
1 minute.
Any questions?
Okay.
Yeah.
If anybody has questions for Maria, they can.
Just for the three of you, I suppose, what I'm hearing is from Maria that very much a whole food plant-based diet is a preferred diet. And what you're trying to do is really take out one part of that product and market it, because that's where you're going to make money. But it's important, I think, to think about the community and what they're eating, and really you're fueling ultra-processed vegan foods.
Well, one reason why we chose dry fractionation is that it's not ultra processing. So what I'm personally interested in much more than protein is fiber, where there's clear lack, especially in Australian diet. So we should eat 30 grams of fiber per day, and nobody's doing that. But people want to, they consume these products. So what we can do is try to make them as healthy as possible. And then of course, when we go to pulses, like John said, there are anti-nutritives, but there are also FODMAPs. So there's, for instance, people with irritable bowel syndrome cannot consume pulses. So you have to figure out other ways of dealing with this.
I'll just do a little ad there as well. I mean, I kind of take the point, but I think the opportunity though is to, because we've failed miserably in our policy of educating people about the dietary pyramid. So we need to continue to do that, but we've got to make convenient foods healthier. So the idea is to not, just mimic meat, but once you've got that system, I mean, I don't think there's anything per se unhealthy about processing. The objective would be to actually build nutrient dense products and make convenient foods healthier. I mean, I think that's the promise. But yeah, I mean, it's we don't equally. It's a diet thing, so we don't. We still want to encourage people to eat fresh fruit and vegetables. But the opportunity is once you've got a delivery platform that's convenient and scalable, etc., then you could take it beyond, the nutrition of meat. You've then got a platform you can deliver new nutrients. Yeah.
Yeah. Yeah, and that's of course our target as well, and we also work together with Helen, so she will keep us on the track. But about the wet fractionation, so John didn't mention it, dry fractionation only works with seeds, because seeds have these granules of starch and protein. If you have green material, like anything, like algae or anything, you cannot dry fractionate it. You have to use wet techniques. And that's one more reason why we want to have all the tools of, that we can have to choose the suitable one.
I just want to answer Bob Gibson here on the line. Good to see you, Bob. Thanks for joining. Yeah, great question there about, kind of what's differentiated here and how do we distinguish from activities with CSIRO? I think the answer is we need to be collaborating, not just with CSIRO, but with other organisations. We've got Michelle Colgrave on the line as well. She's running the CSIRO mission on protein, and we didn't have time today to include her in this one, but I think the one of the objectives is to set up a broader workshop and to make sure we are collaborating. In terms of what we're differentiated here is clearly, we've got the dryland agriculture system, we've got the agronomy side, we've got the fava bean breeding thing, we've got a fantastic food business. So I think we need to kind of work out then what really we need to do from a South Australia perspective. But, we need to have a team Australia. This is a big opportunity for Australia. So we need to have a collaborative Team Australia approach as well. Thanks, Bob.
Okay.
I think we're out of time.
Yeah, we are out of time. So thank you very much for attending, and thank you for all the people on the webinar. We had a fantastic attendance today. Next week's seminar is by a member of the SARDI entomology unit, Rebecca Hamdorf. And if anybody would like to speak during this year, I've got plenty of vacancies from July on, so I'd love to hear from you if you'd like to present your research. So thank you everyone for attending.
depending on amount it is.